Hi, everybody. Thank you so much for joining us. This is the August 2025 monthly seminar on physical genomics. My name is Benjamin Keene here at CPGE, the Center for Physical Genomics and Engineering at Northwestern University. We're really excited to have our invited speaker today, Professor Travis Craddock. Dr. Craddock is an associate professor in the Department of Biology at University of Waterloo and the Tier 1 Canada Research Chair in Quantum Neurobiology. His current research combines quantum physics, biophysics, and neuroscience to investigate quantum processes in neuronal cytoskeletal proteins with the goal of developing novel diagnostics and therapeutics for neuroinflammatory illnesses. He got his bachelor's from University of Guelph in physics and his master's and PhD in physics from University of Alberta, where his graduate work focused on subneural biomolecular information processing and nanoscale neuroscience models of memory, consciousness, and cognitive dysfunction in neurodegenerative disorders. Almost nailed it. Dr. Craddock, thank you for sharing this fascinating work with us today. We really appreciate your time. Just a quick note for our audience, we do the question and answer at the end of Professor Craddock's talk. So if you think of questions while he's presenting, you can type them into chat at any time, and we'll just go through them at the end, or just wait till the end and turn on your mic or type it in then. If you want to learn any more about the research and training programs here at CPGE, you can visit us at physicalgenomics.northwestern.edu, or we've got a video archive of talks and seminars on our YouTube channel, or you can follow us on Twitter, X, Blue Sky, and Instagram. So that's that. Professor Craddock, at this point, I think we can turn our Zoom floor over to you, and we can get going. Thank you so much once again.
All right, thanks very much. You guys can hear me okay?
Yeah, sounds good.
Perfect. All right, I'm going to be talking to you about some of the work I've been doing here since I joined up here at the University of Waterloo. Prior to this, I was down at a university in Florida where I was looking at neuroinflammation and neuroinflammatory diseases. But since joining up here, I have joined my PhD work in the field of quantum biology with my work that I did down in Florida and trying to use quantum biology to try and understand neuroinflammation. I'm going to be talking about an area that we've been looking at specifically, spin dynamics in relation to cytoskeletal polymerization. And then I'm going to tie that to gene regulation right at the end, at the tail end, because it's a little bit different than I think most of the things that you guys are looking at down at the CPGE. There we go. Okay, so like I said, I started this lab up in May 2024, and with the prime goal of using these quantum biology approaches to improve health for neuroinflammatory illnesses. My lab's been growing since we've got up here. The work I'm going to be talking about today is primarily done in collaboration with collaborators down at Nova Southeastern University, as well as the University of Calgary here in Canada, and it's been primarily funded through the Canada Research Chairs program. The work that I'm going to be specifically talking about has been done by my research associate, Hadi Zedhagigi, and my master's student, Caleb Sigüenza, and a little bit of work by Isabella here towards the end. So overall, I'm going to just give you a brief overview of what neuroinflammatory illnesses is and what I'm talking about. What I'm talking about when I refer to quantum mechanisms or quantum mechanics, and then what is quantum biology for those of you that may not know. And then I'll get right into the meat of it, specifically a broad overview of what nuclear spin effects are in biology, and specifically a mechanism called the radical pair mechanism, which is the leading candidate for weak magnetic field explanations in biology and nuclear isotope effects. And then our current work on using this formalism to understand microtubule and cytoskeletal polymerization in brain cells. And then I'm going to try and make the broad jump from this idea that we've been looking at to where it may be useful in the field of genomics. So a neuroinflammatory illness is any illness that has some sort of inflammation in the brain. So in inflammation, we all know about it. If you get a bruise, you hit yourself really hard on the arm, it starts to swell up, it gets sore, and it gets red. And the reason for that is that it's a protective mechanism. It's your body's immune system that's working to try and clear away debris, clear away any damage, and repair the system. When this comes along, it comes with shifts in the environment of the cells that are going on. Two of the things being an increase in reactive oxygen species, as well as there can be an imbalance of metal ions that goes along with that. Now, there's no difference with the inflammatory mechanism that goes on in a bruise on your arm versus what goes on your brain. The only differences are that the brain is a protected compartment, so it's got its own special immune system, and it's got its own inflammatory response, and it's got specialized immune cells that go on. But the process of inflammation is quite analogous to what's going on in the rest of the body. So when it's short term, it's protective. However, if you prolong this state of inflammation, it can start to lead to damage. And we know that now that any sort of brain disorder, from neurodegenerative illnesses to mental illnesses, they all seem to have a chronic component of a low-grade inflammation, at least a low-grade inflammation that kind of starts off and kicks off the process. And this long-lasting neuroinflammation can start to damage the cells in the long run. But we don't really know how that occurs. We know that immune signaling molecules go on. We know that there's an increase in reactive oxygen species and oxidative stress, which can lead to damage. But the exact mechanisms of how that metal ion imbalance or those reactive oxygen species actually damage the proteins within the system is not quite well explained. And so we're trying to understand how this change in cellular environment leads to destruction of protein scaffolds in brain cells, and how that then contributes to things like neurodegeneration. So this is a depiction of a quite famous neuroinflammatory or neurodegenerative illness, Alzheimer's disease, and it's quite applicable to the area that we look at in the cytoskeleton. So within your brain cells, you have the long axonal processes and the dendritic processes. And these are all supported by a scaffold, a protein scaffold called the microtubule cytoskeleton. And it's a regular protein structure that's stabilized by these molecules called tau. Now, in Alzheimer's disease, there's some sort of dysregulation that leads to the microtubules falling apart and these tau molecules clumping together and forming these tangles called neurofibrillary tangles. But what ends up happening in this case is that this microtubule structure falls apart, and then the neuron starts to wither and eventually dies. And so there's two leading ways to think about this. Is one, the tau falls off and then the microtubules disintegrate because it's no longer being stabilized, or the microtubules fall apart, then the tau falls off and it starts to form in these clumps. And so we're trying to tease apart, what came first, the chicken or the egg, and how do these things like oxidative stress and metal ion imbalance contribute to the destruction of these proteins. Now, because reactive oxygen species are primarily radicals with a free electron, and metal ions have nuclear spins and that can interact with protein molecules, we're trying to understand those interactions from a quantum standpoint. So what do I mean when I'm talking about quantum? So quantum mechanisms are things that occur at the very, very small scales of matter. So we're talking about at the level of very small molecules, atoms, or subatomic particles. And so at that scale, things don't follow regular Newtonian classical physics. They follow what's known as quantum physics. And here, this is where the description of what we particularly think of as particles starts to break down, and we really have to start treating these things in terms of waves. So one of the things at the level of quantum physics is that every measure is not continuous, is that certain physical properties are quantized and discrete, and there are unique quantities and qualities of the system that have no classical explanation. So while you may have things like position and momentum, there's things like spin, which does not really have a classical analog. We like to refer to it as an angular momentum, but it's purely a quantum mechanical property. Again, when you're looking at these very, very small particles, or these very small entities like electrons and subatomic particles, you don't want to treat them as little billiard balls moving around. You want to treat them more as waves. They can be delocalized. They can be in several places at once until a measurement's made. And so we need to apply a wave-particle duality description to describe them. Certain certain properties, you can't know two things at the same time. So for example, we quite well take advantage of the fact that we can know both our position and our direction of movement at a given time. When you get down to these smaller level scales, you have to trade off one for another. So there's certain properties like position and momentum, or position and velocity, that we cannot know with complete accuracy at the exact same time. So we can know where we are, but not where we're going, or we can know where we're going, but not where we are. And this is kind of captured in a joke about Heisenberg. Heisenberg's a forefather of quantum mechanics. And the joke goes that he was driving along in a car, and a police officer pulled him over, and he came up to him and he said, "Heisenberg, do you know how fast you were going?" He says, "No, but I can tell you that I'm right here in the car." And so that's kind of an illustration of this uncertainty principle. But there are other properties as well between energy and time, and other non-commutative properties that you can't know both at the exact same time. Quantum entanglement is another quantum property where two entities that interact at some point become entangled, and then you can take them far apart. And as soon as you measure one of them, you can learn information about the other particle instantaneously. So it seems to be some sort of spooky action at a distance in the way these particles interact, but it is something that these subatomic particles can experience. And then finally, quantum tunneling, where if you have a subatomic particle in one location, let's say it's in some sort of well, or within a room, and there's a barrier which it cannot pass between. In a classical description, you would not expect that a particle within a room could then pass through a wall. Whereas at these subatomic scales, these particles can actually tunnel through the otherwise impenetrable barrier from a classical standpoint. And so because of these properties, there's certain ways that we have to treat these particles at that very small scale in order to understand the dynamics of how they behave properly. So how does this apply to biology? So quantum biology is the application of these rules and the description of subatomic particles, or bio-related molecules and atoms, and how these quantum mechanical properties can describe behaviors that we see in biology that can't be described accurately by classical laws. The three areas that are hallmarks of quantum biology have been light matter interactions, such as in things like photosynthesis or in vision. In this case, the efficiency at which a photosynthetic light harvesting antenna transmits the excitation from a photon of light from the top of a leaf down to a reaction center. The efficiency at which that occurs hasn't been well captured by the way the classical description of the excitation hopping between individual light sensitive molecules called chlorophyll. So they have appealed to treating that excitation as a collective excitation shared across multiple light harvesting sensitive molecules. And in these descriptions, they can get better estimates of how that excitation moves, and the speed and efficiency at which it transports. Another area that has been looked at is electron and proton transport. So anytime you have an electron or a proton, which is a subatomic particle that's moving within a biochemical reaction, if you treat it classically, you may not be able to account for things like tunneling. And it's been shown specifically in enzymatic reactions is that the speed and efficiency which those occur can be captured better when you account for those electrons and those protons tunneling through the protein, as opposed to having to go over classical barriers. And then finally, the area that I'm going to be talking about is these magnetic field effects. So this is captured primarily in the field of avian magnetoreception or avian navigation. And so there are certain migrating birds that seem to be able to sense Earth's magnetic field. And Earth's magnetic field is a very, very weak magnetic field on the order of 50 microtesla. And it's not well explained by classical physics uh, how a system could detect those very, very weak magnetic fields, because the energies that are involved with that are well below thermal noise. And so they've appealed to this description of radical pair chemistry to try and describe how a bird or how a biochemical system could sense these very, very weak magnetic fields. And so they appeal to nuclear spin. So like I was talking about before, this is a purely quantum property. But when you're talking about the nucleus of an atom, so not the atom itself, not the electrons, but the actual nucleus of the atoms, the protons and the neutrons, depending on the number of protons and neutrons within the system, is that this nucleus can have a spin, which can be treated as some sort of angular momentum, which has a direction. And when in the presence of a magnetic field, this spin, which is sensitive to the magnetic field, can precess if it's not aligned with that magnetic field. And so if you change the isotope of the atom, or you add a neutron or several neutrons into the nucleus of this atom, you can change the overall spin of the system and the way that it would precess in the field. And classically, these atoms, if you add in another neutron, it should have no effect from a standard biochemical standpoint, because you're not altering any of the electrons within the system. And classical chemistry is all based on electron reactivity. So changing the nucleus and the nuclear spin should not have any bearing on the chemistry. But there's been evidence of isotope effects in biology. So one is that they were studying lithium, and there's two different versions of lithium. There's lithium-6 and lithium-7. One of them has one nuclear spin and one of them has another. And they were feeding it to animals that were they're rodents that were rearing pups in a model of depression, and lithium, or a model of a bipolar disorder and depression. And now lithium is commonly given in bipolar and depression to alleviate those symptoms. And so they gave it to these model animals, and they found that one version, I think it was lithium-6, the animals, behaved normally. They improved their pup rearing, but when they gave them lithium-7, the mothers were neglectful of their pups, and they actually started eating them in some cases. Now, this is somewhat anecdotal because we're looking at the level of animal behavior, but it seems to be evidence of an isotope effect in biology. More recently, again, in rodent models, they were looking at the potency of xenon, which is a noble gas. If you inhale xenon gas, it acts as an anesthetic. And by changing the isotope of xenon and the nuclear spin of xenon, they found differing dosages of xenon were required to anesthetize the animals. And then finally, there's been work that shows that by replacing the magnesium in ATP synthesis with an isotope that has a nuclear spin, is that the rate at which ATP is synthesized by enzymes is increased or decreased depending on the isotope involved, as well as the presence of weak magnetic fields. So these small evidences are pointing towards isotope effects and magnetic field effects based on nuclear spin. But how can this occur? What is the mechanism by which this could possibly occur? And so what they've appealed to is this idea of what's known as a radical pair. So if you have a chemical reaction, you have two chemicals that are coming together to form a reaction. And when they interact, if they're sharing an electron, so an electron from, say, a donor is given over to an acceptor molecule, at the point where that electron is being donated, you now have an unpaired electron on the original donor, an unpaired electron on the acceptor. And those are two then radicals because they have unpaired electrons. And those two radicals, or the radical pair that is there, the electrons that are being shared can have spins, either in a singlet conformation where they are anti-parallel, or in a triplet conformation where they are parallel. Now, the singlet conformation, this is preferred. This is readily allowed, and it allows the chemical process to complete down to an end product. However, the triplet formation is generally forbidden. However, it can be flipped over, and it doesn't react very well. So it either will take a very, very long time to reduce down to that end product, or it can go off to form other chemical end products that are completely unrelated to the original chemistry. Now, it can cycle back and forth between this singlet radical pair or this triplet radical pair formation, depending on internal kinetics, as well as the influence of external factors. And one of those external factors can be an external magnetic field or the presence of a nearby nuclear spin. And so it's been shown that in these chemical systems that you can drive this system either into the singlet state or the triplet state, effectively changing the rate at which these chemicals recombined to the end product, and you can then therefore change the rate of the chemical reaction. And this has been well established, particularly for a flavin-based mechanism in cryptochrome. And within the avian radical pair mechanism, this is how it's envisioned: is that inside the eye of the bird, at the back in the retina, you have your rods and cone cells, and within the rod cells, in between the discs of the rod cells, there's a molecule called cryptochrome. And within cryptochrome, there is a flavin molecule, which is light sensitive. And when a photon enters the eye and gets picked up by the flavin within cryptochrome, it excites from the flavin ground state to an excited state, and then it can then take an electron from a nearby tryptophan or group of tryptophan molecules. And therefore, the tryptophan and the flavin then form a radical pair. And it can cycle between the singlet state and the triplet state, depending again on external factors. And it can change the speed at which the chemical reaction occurs of the way that cryptochrome is responding to that initial photon excitation. And in order for that photon to be picked up in the rod cell, cryptochrome has to complete its reaction. And when it does, it then triggers a chemical cascade, which then causes the neuron to fire, which gets picked up as an optical signal within the bird's brain. And so in the presence of a magnetic field, it will change this in one direction or another. And this is what they believe affects the excitability of neurons in birds' eyes, and therefore gives it the ability to physically see or sense the Earth's magnetic field and allow it to migrate. Now, this is one specific mechanism in cryptochrome, but another area that I had mentioned was this radical pair mechanism in ATP synthesis. So anywhere that a magnesium ion is present, you could potentially manipulate that by changing out the magnesium ion, which is in a standard form, has no nuclear spin. But if you replace it with magnesium-25, which has a 5-half spin, you can then start to see if any system is sensitive to a magnetic field in the presence of that magnesium. And so there's been some Russian groups that have looked at this in terms of ATP synthesis, and they find that in the presence of this nuclear isotope of magnesium, that you can increase the yield of ATP by 2 to 3 times. Just by adding in that isotope of magnesium. And then you can further increase that by the application of a weak magnetic field on the order of millitesla. There's been other groups that have looked at this, and they've looked at it either in vitro and other in vitro systems, as well as in bacterial systems, and found that bacteria in the presence of weak magnetic fields with this isotope, that their growth rate has increased. But if you look at where magnesium actually can affect, there's multiple systems within the body where the magnesium potentially could play a role in mediating weak magnetic field effects. One is ATP synthesis, and then what I'll appeal to later is magnesium's role in processing of DNA and RNA. Now, where I'm interested in this is I'm primarily interested in looking at the cytoskeleton. So, like I had said, inside brain cells, you have these protein filaments called microtubules. They're made up of the protein tubulin, which is a heterodimer made up of alpha tubulin and beta tubulin. And these proteins stick together. They self-assemble when they're in solution into long strands called protofilaments. And then these strands stick side by side and wrap around to form a hollow tube called a microtubule. Now, in brain cells, these form regular patterns of aligned bundles of microtubules within the axons or aligned anti-parallel microtubules within dendrites. But in regular cells, in any other cell primarily besides neurons, they're highly dynamic and they change shape, and they are also responsible for forming the mitotic spindle during cell division. So they are a very fundamental element of the cell, and they play unique roles compared to whether they're in dividing cells like in cancer or in brain cells, which are fairly stable. But within all cells, they're responsible for maintaining the morphology. They form scaffolds in which motor proteins can walk along them to deliver intracellular cargo. If a cell is moving, they help guide the cell. In brain cells, they anchor ion channels, and they've also been shown to have potentially roles within signal transduction, either mechanically or by other means. Again, coming back to these magnetic field effects in the cytoskeleton, this is why we're looking at them, is because when they fall apart, you get neurodegeneration, but they're also disrupted in other neurological diseases. So in major depression, in bipolar disorder, schizophrenia, Rett syndrome, Fragile X syndrome, they've all been shown to have a central role. Their dysfunction has all been shown to have a central role in any sort of nervous system disorder. So if there's any sort of way that we can manipulate these, we're trying to look for that, for any new way beyond a chemical means. So the way that these proteins assemble is that within tubulin, there are two sites where there's a GTP molecule bound. One is between the interface of the alpha tubulin and beta tubulin monomers, and that does not get altered at all. So this is known as the non-hydrolyzable GTP molecule. It never changes, it never releases its phosphate group, and it always stays in GTP form. But this one here on the end of the beta tubulin is bare to the environment. So when tubulin with GTP bound to it at this beta tubulin site is what we call GTP tubulin. When it attaches to the end of a microtubule, causing the microtubule to grow, it'll stay there for a period of time, but eventually it will hydrolyze. And when it hydrolyzes, it goes from being GTP tubulin down to GDP tubulin. Now, as long as a microtubule has a cap on the growing end of it of GTP tubulin, it stays in the growing phase. Now, GTP tubulin, when it does hydrolyze, it bends. The tubulin actually will bend after that hydrolysis, and it puts enormous strain on the microtubule. But as long as that cap is there, the microtubule will stay intact. So I like to think of it as the plastic on the end of your shoelace. So that GTP cap is the plastic. And if it wears away, so if eventually all of that GTP tubulin hydrolyzes and no more is added to it, you can think of it as the plastic coming off the end of your shoelace. And when that happens, your shoelace starts to fall apart and it falls, it breaks down. And that's what happens in the microtubule. When you lose that GTP cap, there's a catastrophe and the microtubule unravels and starts to fall apart. If, however, you start to add GTP tubulin back to the end of it, you can then rescue it and it will start growing again. And so within dividing cells and more malleable cells, these microtubules will go through these phases of growth and shrinkage called dynamic instability. And it gives microtubules their labile nature, which can allow cells to explore the environment, and it can allow it to form and unform the mitotic spindle and so forth. But in neurons, this is highly stabilized by associated proteins that anchor the microtubule to prevent it from falling apart. But because it involves the hydrolysis of this GTP, which is a magnesium-dependent process, so magnesium helps to coordinate the two end phosphates for a water molecule to come in and cleave off that phosphate, we can see if there's any sort of isotope effect or weak magnetic field effect influencing this by substituting this magnesium for a version or an isotope of magnesium that has a nuclear spin associated with it. And so that's what we decided to do. What we wanted to do is we took enriched stable isotopes of natural magnesium, which is primarily made up of magnesium-24, of magnesium-25, which has a spin. Magnesium-24 has 0 nuclear spin. Magnesium-25 has a 5 nuclear spin. And then another stable magnesium isotope, magnesium-26, also has a 0 nuclear spin. And we made up a tubulin polymerization buffer with identical reagents, identical pH, only switching out those isotopes. We wanted to be very careful that we kept all the environments the same, where we were only altering the actual magnesium isotope. We then polymerized tubulin in the absence or the presence of a weak magnetic field. So we would take the assay and we would place it inside of a Helmholtz coil, which is an electrical coils that when you pass current through them, it generates a stable magnetic field between the two coils. And so we would place the different versions of the tubulin polymerization buffer with the different magnesium isotopes within this Helmholtz coil setup. Well, we accounted for the temperature of the system. So we accounted for changes in temperature due to the heating of the coils. We accounted for the pH of the environment. We made sure that everything was stable. All the magnetic fields were the exact same. And then we measured the polymerization of the tubulin via an optical density measure. So we pass laser light through it, and we look at how much the light is diffracted. When tubulin is unpolymerized, it will not diffract the light. But when in microtubule form, they are large enough to actually diffract the light, and you can therefore monitor the way that the microtubule grows in the presence of these different environmental changes. And so what we found was, so these are our conditions. So this is natural magnesium, magnesium-24, no spin, magnesium-25 with spin, and magnesium-26 with no spin. Field off is the blue condition. Field on is the pink condition. There was absolutely no significant change between any of the conditions, except for magnesium-25 in the presence of a magnetic field. And you can see here it jumps up by about 4.5 times what the regular rate was, because everything here is normalized to magnesium-26. So we analyzed this, two-way analysis of variance, found a significant change, Tukey-Kramer post hoc analysis, that only the magnesium-25 in the presence of the field is where the change was. And this seems to suggest that there is an isotope effect, a weak magnetic field effect that is dependent on the presence of this non-zero magnesium isotope. We wanted to make sure that there was no other conditions, so we compared against heating. So we looked at the temperature variation across the individual wells and plotted that versus the actual optical density measure per well to account for any differences by being closer or further away from the edge of those coils. Between runs, we would cycle the location of the different magnesium isotopes. So in one given run, magnesium-25 may have been closer to the coil, whereas the next run, magnesium-26 would have been closer to the coil, and so forth. And we did this multiple times to try and average out that effect. And we found no significant contribution from temperature variation. Then we looked at any sort of minor field variations. So we're looking on the order of a difference of about 20 microtesla between wells. But again, we would cycle the solutions between these different locations on the plates between runs, and we found no significant correlation between the optical density measure changes and the differences in magnetic fields. So we're ruling out that it's due to any sort of temperature variation. We're ruling out that it's due to any sort of magnetic field variation. We do note that there is the presence of some impurities. So the magnetic isotopes themselves have a small impurity with them. We can't get them 100% pure. So any sort of paramagnetic ions like iron or manganese or so forth may influence the system. But the concentrations of these are on the nanomolar range when we're looking at the system. So we're also ruling out the contribution from any impurities. So we're taking this as a real effect, and we're trying to understand, what is it that is occurring. So we built models of tubular polymerization. So it's a simplified microtubular polymerization model built on ordinary differential equations. It's standard used in the field. And we account for the difference in magnetic field and magnetic isotope effects by modifying what is the depolymerization rate. So tubulin polymerizes to microtubules with an on rate, or the polymerization rate on. And then microtubules, if that GTP hydrolyzes and there's no GTP cap, the tubulin can fall off at a depolymerization rate. Now, because we suspect that we're altering that hydrolysis rate of GTP in the system, we're accounting for the change in polymerization based on modifying this depolymerization rate. And so we determine what our change in the depolymerization rate is by modifying this depolymerization rate based on the triplet yield of those radical pairs. So we built a model of spin density description of the radical pair using a standard method that's used in the field, using a Liouvillean superoperator, standard coupling rates and everything like that for the magnesium, as well as we were agnostic to what the form of the radicals were. So we're not claiming to know what radicals are formed in the system. It may be something that goes on during the GTP hydrolysis. It may be the presence of a radical oxygen species in the system that we're not accounting for, but we used a agnostic form of what the radicals were, and then varied it, used a machine learning algorithm to try and determine the optimal parameters of the radicals to reproduce our experimental results. And so using this system, we found that there are a subset of parameters that are consistent with the biochemistry that we're looking at that can recapitulate our experimental results. So here with the points and bars, those are experimental results, and the dashed lines are different parameter sets that give us, something that is comparable to our experimental results. And so we suspect, or we're working under the assumption now that there is a radical pair mechanism that's at play here during tubulin polymerization, but we're not 100% sure what that radical is. We've looked at predictions of this. So at this point, again, we're only looking at two conditions: the geomagnetic field, or the ambient magnetic field, and the application of a 3 millitesla magnetic field. We are currently looking at other points along the curve. These are predictions from our models of what the microtubule density would be under varying magnetic field conditions, as well as the isotope concentration. And we find that even in the absence of the magnesium-25, we would expect to see a magnetic field effect, even though over the range that we were looking at, it was quite small. However, if we were to increase this to higher and higher magnetic fields, we would potentially be able to drive down the polymerization rate, which again is enhanced in terms of it's first increased in the presence of magnesium-25, but then over an increase of magnetic field, that would drop. And so we're trying now, right now, to experimentally compare against this and validate this by looking at other magnetic field strengths. One of our problems is that our plate reader has currently broken, and we're just waiting repairs. So stay tuned. Now, where else might this occur? So magnesium, like I said, is primarily it's a very essential ion in a multitude of biological pathways and biological functions. FAD, flavin, has also been implicated to be involved in these magnetic sensitive reactions. And so my co-op student, Isabella, came up with this idea that if you were to do a magnetic field experiment, and you were looking at the level of the cell as opposed to looking at an isolated biochemical reaction, we may not be able to tell what's driving the magnetic field effects, because there's so many different pathways and so many different reactions that are involved. So what she did was she took two candidates, magnesium and flavin, and used a bioinformatic approach where she went through the protein data bank and pulled out any proteins that were known to bind either magnesium or flavin, and then used those proteins to do an overrepresentation analysis on known biological pathways to try and identify which pathways are highly dependent on either magnesium or flavin. And the idea is that if you can identify pathways that are unique to either magnesium or unique to flavin, and you were to run a cellular level experiment with a magnetic field, uh, and you were to see a change in a pathway that was either primarily or specifically magnesium dependent or specifically flavin dependent, you could indicate whether it was a flavin-based mechanism or a magnesium-based mechanism. However, if it was one of the pathways that you saw where both of those cofactors are involved, you would not be able to determine what was mediating the magnetic field. And so she went through this and then pulled out all the potential biological pathways that were either magnesium dependent or flavin dependent, clustered those based on similarity, and found that magnesium, as expected, was primarily involved in things like neurodegeneration and mitochondrial bioenergetics, ribosome function, DNA repair and cell cycle repair, RNA processing for ribosome function, whereas flavin was involved in mitochondrial redox cluster damage, or oxidative stress and detoxification in the citric acid cycle. And so she identified these individual pathways that we could potentially start to look at to try and identify, where are we seeing other potential magnetic field effects. And it's in these pathways or in these systems that we could potentially start to look at other mechanisms that are driven by weak magnetic field effects or by magnesium isotope effects, that I thought might be interesting in terms of genome regulation. And right now, we haven't had any push in that direction, but it's something that we would like to test. So we're looking for individuals that might be interested in doing this. We have the techniques for or the protocols for setting up the isolated isotope systems, as well as the magnetic field systems, but we don't have expertise in genomics or any sort of DNA or RNA processing type of fields. So if anyone's interested, my door is always open, or my email. Send me an email. You're not going to walk here. Okay, so in summary, the radical pair mechanism is a leading candidate to explain weak magnetic field and isotopes effects in biology. It's been established for the flavin mechanism in cryptochrome, but it remains to be seen if it's viable for other biological systems. Microtubule polymerization, we see that it could be sensitive to weak magnetic fields, and it's an ideal system to test this magnesium-based isotope magnetic field effect. In doing so, we found that there was a isotope-dependent weak magnetic field effect. We rule out any sort of kinetic isotope effect because it can't be due to an increase in mass by going from natural magnesium or magnesium-24 up to 25, because when you increase the mass again to go to 26, that effect goes away. So the only thing it could possibly be would be the nuclear spin. There's no evidence of a pH dependent mechanism because we prepared all the buffers in the exact same pH. There doesn't seem to be any temperature or magnetic field variation driving mechanisms. The amount of impurities does not seem to be enough to cause the degree of change that we're seeing in the presence of the magnetic field. So it seems to indicate that a radical pair mechanism may be viable, but we do not know what those radicals are. We're trying to come up with ways now to test what the actual radicals are involved. Genome operations may be manipulated by similar mechanisms, but it's something that needs to be tested. And so therefore the investigation is ongoing. Here's a list of references that were included in the talk, and I'm open to any questions. Thank you.
Thank you so much. That was amazing. Um, so yeah, as I said, feel free to just type questions into chat and we can take them as they come in. Um, if you'd rather turn on your mic or hold up your Zoom hand, feel free to do that too. Looks like we've got one. Travis, do you want to just read the question and then?
Yeah, so the question says, what would be the role of magnesium spin in the NMDA receptor? It is blocked by magnesium until the membrane of the neuron is depolarized. I am not sure. I mean, there could be a role for it there. I'm not as familiar with what the role of magnesium is in the NMDA receptor. The key for this to occur, at least in the radical pair description, is there needs to be some sort of chemical reaction. So it's in the movement of the electrons, in which the electron is shared such that there's two radicals that are formed. And then it's the nuclear spin of the nearby magnesium that would influence the way that that radical either recombines or stays in the radical pair formation. If there's some sort of chemical reaction that goes on, which I'm not sure if that occurs in that system, if it doesn't, then I wouldn't suspect there was an effect of magnesium. But if there's like a reaction from something like a reactive oxygen species that's attacking NMDA, so if there's NMDA damage by reactive oxygen species, that magnesium may play a role in affecting that. There's a follow-up question to that. Another question. Aromatic amino acids like tyrosine and tryptophan appear to have spin inside magnetic field. What could be the implication in protein function? I'm not sure what you mean by they have a spin inside a magnetic field. If whoever's asking that, if you could elaborate on what you're asking, I'm not familiar with that. When you say a spin inside a magnetic field, are you referring to they're responsive to the magnetic field, or their excitation is responsive to the magnetic field? I'm not sure what you're getting at there. Another question here. The spliceosome is magnesium dependent. What would be the expected results here? Again, it's all dependent on what. So magnesium plays a structural role in protein formation or in I think in nucleobases. Whenever there's some sort of role for magnesium, if it's just an anchoring role where it's just to coordinate nearby residues, then I would not expect there to be an effect of changing out the magnesium or the application of the magnetic field. However, if it's nearby a chemical reaction, so in the case of ATP synthesis or hydrolysis or GTP synthesis or hydrolysis, the magnesium is there. It serves to coordinate the phosphate groups, but because it's nearby when that chemical bond is broken, because there's movement of electrons in some cases, then you're going to be able to influence the way those electrons are being manipulated. And so in the spliceosome, I'm assuming that there's a splicing going on, and if there's some sort of bond breaking or bond addition, then you may be able to change the rate at which those bonds are either broken or they're added, or sorry, where they're joined. And therefore you could either slow down the reaction or speed up the reaction. And that's at the base, that's what this mechanism can do, is that in the presence of the magnesium, you can either slow down or speed up reactions that are nearby, and therefore try and tune the speed at which these things occur. Okay, IIRC, I'm not sure what the acronym is, but it says the applied field 3 millitesla was 2 orders of magnitude greater than Earth's magnetic field. Yeah, that's correct. The abundance of magnesium-25 is in nature's only is 10%, and that is correct as well. You could take advantage of this for synthetic engineered applications, but why would we expect these spin effects would have a role in natural evolved biology? So in natural biology, that 10% may play a role. It depends on how the isotopes are distributed. So if it's just a completely uniform distribution and there's no concentration of the magnesium-25, then you may not expect any sort of noticeable effect from just the application of the weak magnetic field. But there have been not in the brain or anything like that, but it's been shown in certain biological systems that their fractionation of isotopes is weighted differently than what you would find in the natural environment. So I don't remember what the system is. I can look it up if you're interested. Just email me. But certain biological systems will hold on to one isotope versus another, uh, changing the ratio that you would observe naturally in the environment. So in that case, if there's a greater propensity for magnesium-25 in biology, then you would see a greater effect of the magnetic fields. Uh, the way I'm thinking about, not in natural biology, but I'm thinking about this more as an engineering type of approach, is that if we could supplement the system or increase the concentration of magnesium-25 in the presence of a magnetic field, then we could start to influence the way these systems work. It's a engineering and intervention type approach as opposed to a natural based approach. Um, how would isotope separation happen in the cell organism? I don't know. I don't know how that would occur. I just know that there are certain systems that change the fractionation, and I think it's an interesting area of how an organism would hold on. For example, let's say it's magnesium. There's no evidence for that. It was another isotope that I remember. But if there is a way for it to sequester one type of, or one isotope, or favor one isotope over another, the mechanism of which that would occur would be very interesting and would be something that would definitely be along the lines of a quantum biology approach. We do have to rule out whether it's based on nuclear spin or if it's due to a kinetic effect, though. So for example, if it is just that biology prefers to hang on to the heavier isotope, then it might just be due to the mass of it, that it's harder to excrete the heavier version of it versus the lighter version of it. And if you excrete the lighter version out easier, then over time you would eventually end up with a surplus of the heavier version. And so in that case, it would not be a quantum effect, it would just be a mass effect or a kinetic effect. But it's an interesting area to look at.
Okay. Well, if there are no additional questions, I think we can call it a day. Thank you so much. This was extremely fascinating. Um, Really cool. We're gonna have this video uploaded on our archive probably by the end of today. Um, so thank you so much for doing this.
Oh, thank
And,
you. Thanks for inviting me. It's always fun.
yeah, we really appreciate you taking the time. Awesome
All right,
stuff.
thanks a lot. All
Okay,
right.
thanks
Bye,
everybody.
everyone. All right, bye-bye.
Have a good one.